Dr. Malik Palmer, who is a National Professional Officer, Drug Resistant TB in WHO Country Office India, will update us on the treatment of drug resistant tuberculosis and support.
Hi friends, today I will take you through the updates of treatment of drug resistant tuberculosis. In the previous sessions, you have heard about the burden of drug resistant TB, the diagnosis, the diagnostic algorithm, and how we would diagnose different types of drug resistant tuberculosis. So for the treatment aspect, the drug resistant TB patients are classified into these broad classes. The first one is monoresistance, which is resistance to any one of the first line drugs. The next one is polydrug resistance, which is resistance to more than one first line anti-TB drugs other than both isoniazid and rifampicin. Next is multi-drug resistant tuberculosis. This is resistance to at least isoniazid and rifampicin with or without other first line drugs. There is also an entity called as rifampicin resistant TB, which has been recently coined, given the fact that GeneXpert or the CBNAT would give you resistance to only rifampicin as one of the prime drugs. And that is defined as resistance to rifampicin irrespective of resistance to any other first line drug. And largely the treatment of multi-drug resistant and rifampicin resistant goes hand in hand. The next class is extensively drug resistant tuberculosis. Now this is MDR-TB with additional resistance to fluoroquinolones, which could be any of these three drugs, ofloxacin, levofloxacin or moxifloxacin, and a second line injectable anti-TB drug, which is kanamycin, overamycin or capreomycin. So there needs to be at least one of the fluoroquinolone and at least one of the second line injectable drug resistant along with MDR-TB to classify a patient as extensive drug resistant TB. And there could be patients with mixed pattern of drug resistance which also are managed on similar lines as XDR-TB. WHO has prescribed certain principles for designing a WHO recommended DR-TB regimen, which has been adopted in the national PMDT guidelines in India. So in patients with rifampicin resistance or MDR-TB, a regimen with at least 5 effective TB medicines during intensive phase is recommended, which includes a pyrazinamide and 4 core second line anti-TB drugs. One has to be chosen from group A, one from group B, and at least 2 from group C. If the minimum of effective 5 TB medicines cannot be composed as above, an agent from group D2, which is the newer drugs, that is bedaquiline or delamanid, and other agents from D3 may be added to bring a total to 5 drugs. This is the principle to be adopted in designing an effective MDR-TB regimen for any patient. The shorter MDR-TB regimen has been recommended in 2016 based on emerging evidences by WHO, and that has been adopted by the government of India, which now comprises of 4 to 6 months of 7 drugs in the intensive phase and 5 drugs in the continuation phase for 5 months. So a total of 9 to 11 month duration of regimen. And if you look at the composition of this, it meets the principles of designing a regimen that we just discussed, which is it has a pyrazinamide in the intensive phase, it has a group A drug in the form of a high dose moxifloxacin, it has a group B drug which is kanamycin, it has at least 2 group C drugs which is ethionamide, clofazimine, and add-on drugs like high dose isoniazid and ethambutol. And in the continuation phase, we would continue with 5 months of high dose moxifloxacin, clofazimine, pyrazinamide and ethambutol. For patients who may not be eligible for a shorter MDR-TB regimen due to reasons of resistance to either fluoroquinolones or second line injectables, or due to any tolerance related issues or contraindicated indication related issues where the regimen cannot be formed as it is designed here, WHO recommends not to disturb the composition of the regimen but to make another regimen or consider another conventional longer term MDR-TB regimen. So for such patients, the conventional regimen for MDR-TB would be a 6 to 9 months of intensive phase with 6 drugs and 18 months of continuation phase with 4 drugs. Again, the regimen design that you see here meets the WHO recommended design pattern. So we have an injectable, which is kanamycin. We have a fluoroquinolone, which is levofloxacin. We have 2 core oral drugs from group C, which is ethionamide and cycloserine, a pyrazinamide and ethambutol added to this. And in the continuation phase for 18 months, we drop the injectable and pyrazinamide and continue rest of the oral drugs. The government of India also introduced the regimen for isoniazid mono or polydrug resistant TB in the year 2016. The isoniazid mono resistance is clubbed with the poly resistance primarily because most of the poly resistance is always associated with any isoniazid resistance, which means an isoniazid resistance can be considered as a surrogate for poly resistance, and it has been clubbed into one single group to give a standard regimen to manage these types of patients. If you look at the footnote, it very clearly says that although we have a shorter MDR-TB regimen or an MDR-TB regimen for a longer term, rifampicin resistant TB patients can also be treated with the same regimen as for MDR-TB. And in the shorter regimen, there is also a footnote for the injectable, where it states that if the intensive phase is prolonged, the injectable agent is only given 3 times a week in the extended intensive phase. Now let's come to a more complex or advanced form of drug resistant TB and how to design regimens for these kind of patients. So patients with rifampicin resistance or MDR-TB, the regimen designs have to be a little different from what it is in the clear MDR-TB patients with susceptible to fluoroquinolone and second line injectables. So here again, if you closely look at the table, we have 6 regimens here. So let's start looking at the composition of the regimens for each of these classes without the newer drugs first and then come to the newer drugs. So let's take the regimen for MDR-TB or RR-TB with resistance to fluoroquinolone class. So here again we have a 6 to 9 months of intensive phase and 18 months of continuation phase with 7 drugs in the intensive phase and 5 drugs in the continuation phase. So let's look at the composition of this regimen. We have a pyrazinamide in it. We have a group A drug in the form of a high dose moxifloxacin. We have a second line injectable that is group B. We have at least 2 drugs from group C. Rather, we see that we have put all 4 drugs of group C into this to make the regimen much more robust and stronger. And in the continuation phase, we would continue with all the oral drugs except for kanamycin and pyrazinamide in the regimen. Now when you compare this particular class with the same class of drug resistance pattern or regimen where newer drug is to be introduced, we are replacing moxifloxacin high dose with a 6 months of bedaquiline. That's the only change that you would see in the intensive phase, whereas in the continuation phase you would see that the high dose moxifloxacin is not there, but rest of the 4 drugs from the core group C drugs are still there. Let's come to the regimen for MDR-TB with resistance to second line injectable. So when you have fluoroquinolone class available as susceptible and second line injectable class as resistance along with MDR-RR-TB, you can use the fluoroquinolone here. So hence the regimen comprises the duration of intensive phase, continuation phase and the total duration remains the same. However, when we look at the composition, we have still a pyrazinamide in it, a fluoroquinolone which is group A and 4 drugs of the group C, which is the all oral drugs. And 18 months of continuation phase is without an injectable but with a fluoroquinolone in it, and ethionamide, cycloserine and linezolid continued. The same type of regimen with newer drug would be more or less the same with the addition of a 6 months of bedaquiline in the intensive phase. Rest of the regimen with the footnotes remain the same. Let's come to the regimen for XDR-TB. Here we have a 6 to 12 months of intensive phase and an 18 months of continuation phase. And if you see the number of drugs in these regimens, they are much more. So if the patient is not eligible for a newer drug like bedaquiline, we would start with high dose moxifloxacin, capreomycin, which is the group A and group B, 4 drugs of the core group C, and a pyrazinamide and ethambutol added to it. And in the continuation phase, we would drop the injectable and the pyrazinamide from the regimen, and rest of the oral drugs would continue. And when we compare this with XDR-TB regimen for newer drugs, you would see that the high dose moxifloxacin has been replaced with 6 months of bedaquiline, and rest of the regimen more or less remains the same, while in the continuation phase you do not have a high dose moxifloxacin, but we continue with the remaining 5 drugs as all oral regimen. Before starting the patient on any regimen, it is essential to evaluate the patient on clinical, bacteriological and biochemical grounds to look for any underlying derangements which may have its effect on the management of the patient. So these are the investigations which need to be done for practically all drug resistant TB patients. But there are some more additional investigations which may be necessary at baseline, particularly in patients where we are considering to use shorter MDR-TB regimen, which has 2 cardiotoxic drugs in the form of a high dose moxifloxacin and clofazimine, or patients who are being considered for bedaquiline, delamanid, new drugs who are also known to have other side effects. So these are the additional investigations which are required in such patients. Follow-up investigations are also important to monitor the progress of the patient on treatment and the safety of the regimen the patient is on. So as part of the clinical monitoring, as well as bacteriological monitoring, as well as monitoring for side effects, a set of scheduled investigations have been proposed for all MDR-TB patients. So a follow-up is to be done by sputum culture or smear microscopy on a monthly basis in the intensive phase, and culture every 3 months in the continuation phase. If intensive phase is extended, if the culture is positive in intensive phase, the extension would continue up to the maximum duration mentioned for each of the specific regimens up to that particular time point. Monthly cultures would continue. Apart from that, clinical monitoring and various biochemical investigations, the frequency of which has been defined in the program. So that's about it as far as the treatment of drug resistant TB is concerned. So friends, to conclude, let me reiterate this point. Recently, WHO has issued a rapid communication based on evolving evidences around safety of newer drugs and a meta-analysis on safety and efficacy of various drugs for longer regimen. And in that, there are changes recommended based on newer evidences, which the Government of India and the National Expert Committee are reviewing it extensively. So in the near future, you may see a revision or a further update based on these recent evidences of the national guidelines, which may be presented to you subsequently. So keep a lookout for that particular thing in the next time.